Welcome to the PowerPoint cells. As you can see on the screen, we've got the building blocks of people and other living things. We start with the cell. A collection of cells is our tissues, and all those tissues make up organs. You can see this man holding his skin. That's a person from the body's exhibit. So we're going to look into cells. So plasma membrane is the outside. So here you go. That's the outside. Cytoplasm is the inside. And the crucial center is the nucleus. So here we have the plasma membrane. All right. So these little guys in here are phospholipids, and you'll see that you have two layers of them. So it's called a phospholipid bilayer. And the phospholipid bilayer is embedded with these proteins, these things coming off of here. Typically, those are sugars that you see coming off. Right? This one not only has sugar, but it also has some fats in it. That's why it's called a glycolipid. But these guys are all proteins. The proteins are embedded in the phospholipid bilayer and allow stuff to come in and out of the cell. So this word semi-permeable is there to tell us that, well, not everything can get in, and that is why it is selective. Protects the cell, it compartmentalizes it. Here's your microvilli right over here. They just increase the surface area of the cell. Okay, this thing is key over here. It doesn't increase the volume, and that's important because the whole reason why we wanted to increase the surface area is because we wanted to be able to get more stuff in and get more stuff out that we were creating. Now, if we also increased volume, then we're creating even more stuff that we have to deal with. So we don't want to deal with more stuff. We just want to have this extra tool. Okay? Some people think that it is mobility related. They think that it is, some kind of cilia or something like that. But it is not. It is not. And we will see that on another slide. Here it is. Here's mobility. This says flagella. I don't know if you can see that too clearly, but that's this guy that's coming off of this particular cell. And then there's cilia, and that's all these little guys that are coming off, okay? Now the main difference between these two is that this acts more like a whipping motor, and it allows that cell to propel itself, whereas these things are more like moving the substance and just kind of more of a surface thing. So this is like, this thing is moving, and this is like the water's moving, okay? Cytoplasm, everything from the nucleus to the plasma membrane. Notice that this one's called a cell membrane, but that must mean that what you're looking at is a cell, which it is. The phospholipid bilayer that had all of those proteins in it, that is a plasma membrane, and that's really the word that we like to use because it tells us something not only about cells, but about all organelles. So this piece here is important. We do interchange cytosol and cytoplasm, but the truth is cytoplasm is cytosol, organelles, and all the inclusions. So here's inclusions. These are non-functioning units, all these little guys that are in here. Not these big ones. These bigger dots are ribosomes, but these little guys that are up in here, those are representing our inclusions, and they generally are just, equated to just junk stuff that's in there, but it is stored nutrients and cell products. The cytoskeleton is designed to help keep the organelles in their places, okay? It also assists with some cellular movements. It is associated with the flagella. Then you have organelles. Let me pick this up a little bit so you can see that. Organelles are all of those things that are inside of the cell, okay, that have specific jobs. So first of all, let's look at that plasma membrane that's on the organelle itself. Remember we were talking about that earlier. Okay. Compartmentalization allows for otherwise incompatible functions to coexist. So think about that for a minute. We're talking about inside of a cell. So imagine inside of your body if your stomach acids were just in there all loose all over the place. So you get the idea. They need to be compartmentalized. Okay. So all those little, as it says here, microenvironments are secure. Okay, centrioles, these guys are kind of neat. I think they look kind of like churros if you're familiar with that Mexican dessert. They always come in pairs. They are going to double when you're getting ready, right before you're getting ready to divide a cell. You'll have one on either side of the animal cell. They'll send out these spindle fibers that will attach to the chromosomes, and then they'll pull those chromosomes apart. The endoplasmic reticulum, often abbreviated as ER, comes in two sizes. Sorry about that. Rough and smooth. It's rough because it has all these little ribosomes that are on it, making it a rough appearance. Smooth, I think you get the idea. Look at this though. The rough is all protein related. Do you see that? Right? It deals with proteins. Now the reason it deals with proteins is because ribosomes make proteins. Right? The smooth, lipids and fatty acids are both associated with, that's the same thing. So smooth is like the fatty acid part of it, or the lipid part of it, and then rough are the proteins. So there's 4 macromolecules. Proteins is one, and then the lipids is another. And so the endoplasmic reticulum is responsible with those two things. The Golgi apparatus. Okay, you got it. Those proteins are made and they have to go somewhere. So the proteins are made, the lipids are made, and those are now going to be modified in the Golgi apparatus, and then they're shipped wherever they're needed. And so what you see here, all these little vesicles, they're going to have, well, this one says incoming, but these guys that are popping off, like right here, this one's popping off, and then there it is. That's one of those enclosed vesicles that has the proteins or the lipids in it. Now what's interesting is all of these little layers actually do, it's kind of like they melt. It's pretty cool actually. Alright, some more vesicles. Alright, there's lysosomes. That's what you see coming from the Golgi bodies. Now they have enzymes in them that are going to digest the macromolecules, which means that they can digest proteins and fats and carbohydrates. Okay, and because they can digest those, it's recycling that material. Right, peroxisomes are also these enclosed vesicles that have enzymes in them that detoxify, essentially. So peroxisomes, hydrogen peroxide, that helps you realize hydrogen peroxide in your body, not so good, and peroxisomes help to break it down. Vacuole is another vesicle. Vacuoles are in mature plants. That is, they're storing all these different kinds of substances. You can see amino acids, sugars, and toxic substances as well. And so when the fluid accumulates inside there, that's going to make the cell wall enlarge, and we call that turgid. So if you've ever, had your carrots and they're nice and crisp, they probably, all of those vacuoles are full of water and other kinds of substances. And then when you get them and they're kind of limp, that flaccid indicates that it has lost some of that water. Okay, why is that not there? And there it is, making energy. Mitochondria and chloroplasts are the 2 ways that we're going to make energy. Alright, there are 2 organelles that are expressly related to making energy. That's the mitochondria and the chloroplasts. You're going to find chloroplasts inside of plant cells and in some protists. And these guys, well, and some bacteria, and they are specializing in photosynthesis. Remember that photosynthesis is where you take sunlight and you add water and you add carbon dioxide and you end up with C6H12O6, which is our sugar, and a byproduct of that is also oxygen. Yay, thank you plants. Okay. Now, the reverse of that is for mitochondria. So mitochondria is what all organisms have inside of their bodies, except, of course, bacteria. Sorry about that. And we eat. That's that sugar, C6H12O6. We breathe in. That's the oxygen. And through that process, our mitochondria can take those things and through a series of reactions produce this stuff called ATP, which is the energy source for all of the work that has to be done. And here it is, the energy movement in cells. Okay, so look here. That's that C6H12O6 that we're talking about. It's set down into its empirical formula because there's no coefficients in front, and they just wanted to show that it's balanced, okay? But organic matter, carbohydrates, same thing. This is just a specific organic matter, plus the oxygen. You can see over here it's a byproduct. Okay, this symbol means yields carbon dioxide and water, and this one needed carbon dioxide and water. Okay, now sunlight is on this side. You have to have sunlight, which happens to be an energy source, and it's interesting that you'll notice over here for respiration you also have energy. Okay, now there's something special about the structure between mitochondria and chloroplasts. Yeah, this is the theory of endosymbiosis. Here are your ribosomes. It's the site of protein synthesis. All these little guys, those are each ribosomes. They are not membrane bound, which means that they do not have that plasma membrane. It's where translation takes place. That's how we read the mRNA code. So that's what you see going on right here. Here is a tRNA. Okay, that tRNA shows up with its own little amino acid, which is right here. And it knows to attach itself right there because this code, this mRNA, says, hey, attach here. And so these guys, these two codes are complementary, and the ribosome helps to attach those two in place. And then as another tRNA shows up, you get this growing chain of amino acids. And this is that process there. The process of going from DNA to RNA to protein is called the central dogma of molecular biology. So the DNA code is transcribed. RNA messages are translated, and then where are we going to put all this stuff? So we're going to stockpile in the cytoplasm, or maybe we'll put it in the ER, and then it'll be packaged and processed and then sent off to the Golgi apparatus. Here's our nucleus. We like this guy. He keeps DNA and its transcription into RNA away from all the damaging stuff that could happen to it inside that cytoplasm. Remember, DNA is very large, cannot leave the nucleus. Here are the various nuclear parts, chromosomes, what they do, chromatin, nucleolus, nuclear envelope. Okay. Nuclear parts again, the chromosome, the chromatin, the nucleolus, the nuclear envelope, and the nuclear pore. So if right here I'm looking at a nucleus, this whole outside would be called the nuclear envelope. Anywhere where there's these little holes, those are the pores, the nuclear pores. That's where the RNA gets out. Remember, DNA is too large, it's not going to go out. All this fuzzy stuff is chromosome, and chromosome is just the tightened up version of chromatin. The very center of a nucleus is called the nucleolus, and if you'll recall, the nucleolus is all about the construction site where ribosomes are assembled from RNA and proteins. Okay, and here you can see that process. You see these little guys in here? Those are chromosomes. So here's the big chromosome. Every chromosome is made out of a strand of DNA that is circled up on these histone proteins and supercoiled into that chromosome. There's various ways that you can study for this. Let me move this down a little bit. There you go. Here's one way. Okay, it's a pretty neat model you got there. Of course, the trusty note card. You could also compare, how is a cell like a city? Study it that way. Creating a PowerPoint, you could possibly take these steps that you have here on this PowerPoint slide and then manipulate them, print them out, and then you can also show how they're all related to each other. And that is that.